Thank you again for joining us for today's research webinar, Pathogen Contaminated Wastewater, Where Does It Go and How to Treat It? It is my pleasure to introduce our presenter, Dr. Adam Birdsall. Adam is a postdoctoral researcher with EPA's Center for Environmental Solutions and Emergency Response. He conducts and manages projects focused on waste management, bioaerosols, and pathogen destruction. These projects include research on bioaerosols, emission mechanisms, pathogen fate, and persistence in infrastructure and waste. Adam has a BS in geology from Wittenberg University, a MS in Earth and Environmental Sciences from Wright State University, and a PhD in Environmental Sciences from Wright State University. And now I will turn it over to Adam. Thank you.
Hello, everyone, and welcome to this talk today. I'm Dr. Adam Birdsall, and I am here. Let's see here. Hold on one second. I need to make sure that everybody can see my slides. Okay, so thank you for coming. I'm Dr. Adam Birdsall, and I'm here to present to you a summary of our recent work here at EPA on pathogen fate and transport in wastewater treatment plant infrastructure using pathogen surrogates. My colleagues for this research are Dr. Matthew Magnuson and Dr. Jeff Szabo. There we go. Okay, so I just want to start off with a brief discussion of why we care about wastewater. To put it simply, it's critical to wide area water security and resilience. From the time water is taken from the environment, is treated, distributed, and used as drinking water to the time that it's collected for treatment at a wastewater treatment plant and discharged back into the environment, there are many threats that can introduce dangerous pollutants that the wastewater treatment plant may or may not be fully equipped to handle. The threats addressed in our emergency response research have expanded beyond terrorist attacks to include accidental events like CBR backflushing, transportation accidents and spills, runoff from decon efforts for chemical, biological, or radiation contamination, naturally occurring events like weather hazards and pipe breaks that happen just as infrastructure ages, and deliberate actions that can have unintended consequences like the use of firefighting foams. What we're honing in on today is pathogens, and we've had a few incidents in recent memory that highlight the importance of pathogen research in wastewater systems. In 2001, we had the Amerithrax incidents where Bacillus anthracis spores, or BA for short, were sent to several US post offices and presented quite a problem for cleanup and disposal. It's also worth noting that anthrax outbreaks can occur accidentally in livestock populations. In 2014, Ebola outbreaks piqued the interest in providing wastewater treatment service information and preventing pathogen spread. And most recently, wastewater testing was used to help us understand the magnitude of COVID-19 spread in communities. Highly resistant pathogens can make their way into the wastewater treatment system at any time. And the problem is that wastewater treatment plant operators, as one of the last lines of defense against those hazards spreading to the environment, don't like accepting unfamiliar wastewaters. To that end, water research and environmental foundations have held workshops to identify industry concerns about accepting contaminated wastewaters. We've been working closely with them, and the projects that follow are a result of consultation with wastewater treatment experts during these workshops. The workshops themselves cover chemical, radiological, and biological decontamination. But as I said, today we're focused on high consequence pathogens. We hope that our research helps wastewater treatment experts meet some of their goals, such as choosing unified sole source guidelines so that everyone's working from the same playbook, characterizing contaminants to take some of that mystery out of those unfamiliar wastewaters, taking measures to protect workers from those contaminants, verifying that the treatments that they've been implementing are successful, and protecting the infrastructure, treatment processes, and receiving waters against future contamination. The work I'm discussing today is being conducted at EPA's Test and Evaluation Facility here in Cincinnati. I'll be talking about three areas of study. The first one is looking at the persistence of pathogens in a pilot scale model, the aeration basin and return activated sludge treatment system. Then I'll talk about some common methods for treating dewatered biosolids and explore how spore forming surrogates might actually survive some of those methods in lab scale. And finally, I'll discuss bioaerosol release mechanisms and our study of the impacts of surrogate type and different media compositions on their release. So now I'd like to introduce to you our surrogates. These are organisms that have some similarities to pathogens, but are not dangerous to humans, so that we can use them as stand-ins or substitutes with less risk of infection. Spores of Bacillus globigi, or BG for short, are a common surrogate for highly resistant spore-forming pathogens like Bacillus anthracis, which causes anthrax. The rod shapes in this photo are the BG in their vegetative state. When they're in an inhospitable environment, they tend to expel much of their moisture from their bodies and pull in their resources to form hard, durable particles called spores until they can find an environment that they like better. That's our white dots in that photo. MS2 bacteriophage is a common substitute for non-enveloped viruses like poliovirus, and Phe6 is often used in place of enveloped viruses like Ebola and nowadays SARS-CoV-2. Those high risks that I talked about and negative optics of a wastewater treatment plant taking on contaminated materials has led wastewater treatment experts to ask a number of critical questions about pathogen survival, like how do they survive in the wastewater system? And by extension, how do they survive that treatment process? What are their survival rates on surfaces? How are they surviving in aerosols? Our research is conducted to provide some answers to these questions and hopefully provide further insight to help mitigate pathogen survival. So now that I've laid some of the foundation, let's get into the research, beginning with the persistence of surrogates that I introduced earlier, and in the pilot scale activated sludge treatment system pictured here. This work was published in PLOS ONE in 2022, and this system is made up of three major parts. To take you through the system step by step, the diagram on the next slide, it will show you from the other side. So we've got beginning, middle, and end, and beginning, middle, and end. So going from left to right, we have our primary clarifier, which is where we simulate the introduction of the pathogen. The aeration basin in the center is split into 6 chambers with coarse bubble diffusers to keep the oxygen levels up. And then the secondary clarifier is on the right, where some of the materials get discharged from the system and some of it gets recirculated back to the aeration basin as return activated sludge, or RAS. We injected a slurry of one of each of the 3 surrogates into the system at a constant rate, usually over 72 hours, and studied how long each one persisted in the system. Liquid samples were collected at regular time intervals from several ports along the way in order to monitor for viable organisms and suspended solids. Dissolved oxygen and ammonia were also monitored. The length of time that the organisms were detected throughout the system was compared against the hydraulic retention time, or HRT, and solids retention time, or SRT. The hydraulic retention time is defined as the calculated time it might take for a particle of water, if we could track it, to travel through the aeration tank. The solid retention time is the same value for a particle of solid material. Bacillus globigi spores had the most interesting behavior. So let me break down this figure because there's a lot here to take in. First thing, the concentration of viable colony-forming units per 100 milliliters of sample are located on the left y-axis here in scientific notation. So you have 10, 100, 1,000, etc. This axis pertains to the blue, orange, and red series. And for these data, it made the most sense to average the data together according to which basin the material was coming from. So all of the samples coming from the primary clarifier are in blue, then the average of the aeration basin, and finally the effluent is the material that came out of the secondary clarifier. Total suspended solids measurements are measured along the right y-axis here in milligrams per liter and pertain to these 2 green TSS series. The light blue field on the left here is the range of possible hydraulic retention times, and the yellow field with the green line through it is the calculated solid retention time with a range of possible uncertainty. BG concentrations eventually return to near their detection limits in the primary clarifier, so for the beginning parts of the apparatus. But in the rest of the system, the aeration chamber and the secondary clarifier, concentrations remained rather high throughout the experiment, long beyond the hydraulic retention time, but even beyond the solid retention time range. This suggests that one of 2 things was happening. Either BG colonizes the system, leading to the growth and production of new organisms from within the plant's infrastructure, or it's simply accumulating. What happened with MS2 will give us a clue that it's actually the former. So for MS2, we have a similar figure here. We did a couple of different tests that I have separated out. One of them had mostly dechlorinated tap water, and then the other one used a synthetic wastewater mixture that includes several chemicals that are designed to emulate things that are normally found in actual wastewater. It would have contained more organic materials, but there wasn't really a distinct difference between the two experiments. But what was significant is that MS2 concentrations in all parts of the system dropped to near their detection limits at or shortly after that possible solid retention time range. Since both MS2 and BG are solids, it's not surprising to see them stick around beyond the hydraulic retention time. But why didn't MS2 stick around beyond the solid retention time the way BG did? The answer? Well, MS2 is not expected to grow in the system because its host organism, E. coli, was not expected to be present. Since it also did not accumulate over time, we deduced that BG wasn't accumulating either, and instead that it had colonized the system and that the MS2 would eventually simply leave the activated sludge treatment system. MS2 was detected in small amounts beyond the solid retention time, but the levels were low and seemed to be within the range of likely uncertainty. V6 was a bit more problematic. One problem we encountered was that we couldn't cultivate it in quite the same concentrations as our previous two surrogates. To keep the detected concentration up, the overall flow rate of the system was slowed a bit. This lengthened the hydraulic retention time and solid retention time, but it didn't really seem to matter very much. V6 made it past the hydraulic retention time without too much trouble, but the concentrations dropped to 0 before the solid retention time range even began. This is because V6 is very highly temperature dependent and could be easily inactivated by water at room temperature. Temperature is important to the viability of all of the surrogates, but V6 is the only one that needs the temperature near freezing to stay viable. The conclusions from this study indicated that even short-term exposure of an activated sludge system to a spore-forming bacterium like Bacillus anthracis that would require oxygen could lead to colonization of the wastewater treatment plant's infrastructure, causing the plant's own system to actually become a source of future pollution. Future work on this topic should include development and testing of approaches to decontaminate these systems after they've been colonized. Secondary wastewater treatments can largely be effective for enveloped viruses. The overall likelihood that an activated sludge treatment system would become source for such a viral pathogen is relatively low. But the treatments that do get applied for a sludge treatment system need to be run for a longer period of time than the system's range of solid retention time after the contamination's been detected. Second study that I want to discuss is our assessment of several methods that are often used to treat dewatered biosolids that have been contaminated with Bacillus glabiji spores. So we're putting the viruses aside for now. We're just going to focus on spores for this one. This work is currently under review for publication, and you can see our lab scale incinerator system here. Dewatered biosolids are simply the solids that remain after the liquid's been removed from the influent material using an apparatus like the centrifuge pictured here. Biosolids are also commonly called sludge, but I'll try to keep referring to them as biosolids here so that it's not confused with the mostly liquid return activated sludge that was being used in the previous study and in the third study. Wastewater treatment plants produce many tons of dewatered biosolids every day, and some may contain pathogens that pose risks to plant personnel and the surrounding community. We investigated three methods in the literature and then at the lab level if it looked promising. We first looked at lime stabilization, which treats pathogens with calcium oxide or calcium hydroxide in order to produce a high pH and temperature condition. However, for this study, our research did not reach the lab level for two reasons. The first reason it didn't reach the lab level is that the calcium in lime is known to actively help to fortify and protect bacillus spores, increasing the risks of future infection. In fact, the Hemsworth paper that I cite here suggests that lime should never be used to treat such pathogens. The second reason is that EPA considers lime to be a Class B biosolid producer, which is designed to reduce pathogen concentrations, but not necessarily eliminate them. For something like anthrax, we wanted something that was a Class A biosolid producer, which would provide more thorough treatment. So for this investigation, lime was out. So we moved on to aerobic composting. This is used for managing manure and dead livestock, which is important for those accidental anthrax outbreaks I mentioned earlier. And then we moved on to fluidized bed incineration, which is widely regarded as one of the most effective treatment methods. In aerobic composting, pathogens are inactivated by prolonged exposure to high temperatures. For spores, this generally requires temperatures to be above 60 degrees Celsius. The drawback to this is that when the temperatures get above 70 degrees Celsius, sometimes we can start inactivating the organisms that help to produce those high temperatures in the first place. So it leaves us with a somewhat narrow window for optimum performance. Effective aerobic composting also needs air, a carbon to nitrogen ratio of about 25 or 35 to 1, and moisture. Large-scale composting often requires forced aeration by either turning the compost over or by pumping air into it. Our lab scale system here was small enough that it didn't need either one of these. Some holes at either end of the composter were enough to keep it from using up all of its oxygen. Moisture becomes important for bacillus spore removal because spores are easier to remove with moist heat than with dry heat. Moist heat damages the spore membrane, while dry heat is mutagenic. This kind of becomes our theme song for this study, so it's important to bear this point in mind moving forward. It's also worth noting that when discussing spore inactivation or removal, we're talking about organisms that have been killed, yes, but it also includes organisms that are simply rendered unable to grow and reproduce. To make the composter, biosolids were mixed with a pine mulch bulking material at a 1-to-1 or 3-to-2 ratio by mass and placed in a 55-gallon drum pictured here. We placed a warming jacket around it to help keep the temperature near that target temperature of 60 degrees. I added BG to some samples of biosolids and buried them in the composter in perforated nylon bags inside cloth sample bags. I also tossed in a vial of BG liquid suspension into the cloth bag as a control for the materials in the composter. So getting into the data here, let's first look at that 1-to-1 mass ratio of biosolids to bulk, starting with the y-axis like I did before. Like in the previous study, this axis is showing the amount of colony-forming units remaining in the sample, beginning at around 10 to the 8, or 100 million colony-forming units. This is for the red and purple series going forward. The right y-axis is for all of the temperature readings that we took that are plotted in grayscale, including measurements in the center, at the bottom, and at the outer edge of the composter. Plus, I plotted the thermostat set point on the warming jacket. Over the course of the experiment, we only removed about 2 log units, or 99% of the BG spores. That sounds like a lot, but as you can see, there's still plenty left to infect another day. One reason we had trouble removing spores was because our experiment struggled to maintain a constant temperature at the target of 60 degrees Celsius. As you can see, we have the warming jacket set all the way up to almost 80 degrees Celsius, and we only have a center temperature of around 40. Others have also reported that localized variations in temperature can cause uneven removal. Another reason was that this composter's material mostly dried out by about halfway through the experiment. We divided our samples into samples that were heat shocked and those that were not heat shocked, hence a purple and a red series here. Heat shocking involves subjecting the sample to 80 degrees Celsius temperatures for about 10 minutes to kill off any vegetated organisms. If the heat shocked samples had a lower yield than their non-heat shocked counterparts, it might be a sign that spores were finding sufficient nutrients to both germinate and survive in the compost. The lack of such a difference in most samples means that either no organisms were surviving, or at least that none of them were living in the compost. We suspected that if BG had germinated, they were probably quickly inactivated by the heat of the composter. A composter with a bit more biosolids was able to retain its moisture a bit better and also had better removal, about 4 log units or 99.99% of the BG spores. That's our red series here. The blue series of control vials showed no significant removal. This tells us that the removal of these spores is not just from high temperature. Either something in the compost or something about the spore's response to it is resulting in their inactivation. We believe this to be further evidence that the spores do find sufficient nutrients to come out of their spore form, and when they do, they're damaged and inactivated by the heat. Moving on to incineration, this is a diagram of our incineration setup. Our fluidized bed furnace uses very high temperatures, about 500 to 1,000 degrees Celsius, to burn the biosolids and kill pathogens. Inside the furnace is a glass tube with a perforated frit. A bed of olivine sand sits on top of the frit, and a compressed air tank delivers air from underneath that filters up through the sand, causing it to churn and boil as though it's made of liquid. When the furnace reaches the target temperature, Bacillus glabiji contaminated biosolids are dropped in from above with our biosolids feeder. Sand has to be mixed with the biosolids samples, though, in order to prevent the pieces from clumping together and bottlenecking in the system. The air that travels up through the bed leaves out this exhaust line and heads over to the impinger, carrying all of the solids, ash, sand, and any surviving organisms. The impinger collects the solids and organisms, and the air then leaves out the exhaust snorkel. Samples were collected from the sand and ash that remains in the fluidized bed, the exhaust tubing, and the impinger. Solids were then suspended in the sterile DI water to extract any living organisms into the liquid where they could be plated. Okay, so first off, these figures are set up in reverse of the previous ones that you've seen so far. Instead of colony forming units that have been recovered, these bars are showing how much has been eliminated, up to about 10 to the 8th or 100 million, which is about how many spores we were adding to the samples. So above this line, we expect that there has been complete removal. The first item on the itinerary was to look at how the temperature affected spore removal. And our findings weren't quite intuitive. Hotter was not necessarily better. As the temperature went up, the amount of BG apparently removed actually decreased. The lowest temperatures that we tested, around 500 degrees Celsius, retained a lot of their moisture during the test and seemed to more easily inactivate the BG. Hearing our theme song yet? The 1,000 degree experiment had lots of survivors, all of them in the impinger, but nothing in the exhaust tubing, which we estimate was because as the system was cooling off, it was still subjecting all of the organisms in the tubing to hot air. Oops, sorry, I wanted one more point there. We checked the ratio of biosolids to sand that we were using in our sample preparation as well. That's our orange and our pink here. And it didn't seem to have a whole lot of effect on the outcome, so we didn't pursue that further. But when we first started this part of the investigation, we believed that biosolids would have a largely similar moisture composition from one sample that we retrieved to another. But this wasn't really the case. The process of inoculating the biosolids with BG actually had to change when the sample had more moisture. Dry samples could be folded, but the wetter samples had to be stirred. The importance of this? Well, those high initial moisture content experiments meant near complete removal from all parts of the system. That's our blue and purple series compared to their drier orange and red counterparts. And again, it's the same thing. Moist heat removes more bacillus spores than dry heat. One of the biggest challenges to making this bench scale incinerator work was how samples could be delivered into the furnace without samples bottlenecking or sticking in the feeder. One way we tried to cut back on the sand that we were using to mix with the biosolids was to put the contaminated biosolids into gelatin pill capsules. This eliminated most of the sand that we mixed with the biosolids, but some sand was still necessary to keep the pills from sticking together once the moisture started to dissolve the gelatin. We also saw few to no surviving organisms, as you can see in our green series here. But there was a problem. These capsule experiments left sandy slag shells in the fluidized bed sample after each experiment. It suggested that the gelatin may not have burned up during the experiments and may have prolonged the sample's exposure to the intense heat beyond what would normally be experienced in the full-scale incinerator. This doesn't really help us solve our problem of seeing what the result would be without added sand, but it does seem to highlight the importance of sample contact time. So now for the overall assessment. The literature suggested that lime may help to fortify bacillus spores. As such, we did not pursue the matter, and it remains an unrecommended approach. However, much of this is still based on assumptions from how vegetative forms of the bacteria use calcium during the process of becoming a spore, so a revisitation of this topic may be in order. Composters could remove much of the contamination, but they struggled to maintain the needed temperatures. In large-scale systems, the maintenance needed to keep the oxygen in the compost can release hazardous bioaerosols. So while it might be an effective method for general pathogen destruction, adding highly resistant pathogens to the mix creates a situation where composting may not be the most ideal solution. As we expected, incineration was the most effective of the three treatment methods, but there were some factors that seemed to maybe increase the risk that pathogens could survive this process. And so we offer these ideas that might be adopted at some point to mitigate that risk. Number one, allow more moisture to remain in those biosolids when they are injected into the incinerator. We're gearing up to take a much deeper dive into this part of the investigation in a follow-up study. Number two, operators could run their incinerator at a lower temperature to allow the biosolids to retain more of their moisture for a longer period of time. And finally, operators can make sure that the place where the contaminated biosolids enters the furnace is as far from that exhaust outlet as possible, thus maximizing the amount of time that it's exposed on its way if it's going to try to make it out that exhaust outlet. So don't use these inlets that are close to the exhaust if you can help it. That brings me to study number 3 in our last stop, bioaerosol emissions of surrogate pathogens in wastewater. Some of this work was conducted at the Air Force Institute of Technology, or AFIT, up at Wright-Patterson Air Force Base, in collaboration with the US EPA. I'll start with a brief overview of some of the work that we did there to give you some background on what we're doing now, and then move on into the ongoing work in this field being done here at EPA. So bioaerosols consist of organisms, parts of organisms, or chemicals from organisms that are emitted by splashing and bubbling. We're mostly concerned with viable organisms. Splashing can emit a wide range of droplet sizes, not all of which are going to be small enough to stay airborne as bioaerosols. And bubbles come from air that's been forced down into the water by either air diffusers or by rotating aeration brushes, like the one diagrammed here from our 2021 review paper. There's not been a whole lot of attention given to the understanding of the factors that affect the mechanisms of bioaerosol release, but we have done some research on how to mitigate bioaerosol release. Since bubbles are the primary method of bioaerosol release that we're discussing in this presentation, it's worth taking a little bit of a closer look at how the aerosols are generally emitted from them. As a bubble travels up toward the surface of the water, it collects organisms and parts of organisms where the air bubble meets that water. When it reaches the surface, the organisms are either in the membrane on top of the bubble or are in the liquid just underneath it. The membrane thins as liquid runs down the sides of the bubble. When it bursts, the membrane breaks apart into tiny droplets. If a droplet containing an organism is small enough, it might become airborne and become a bioaerosol. Then, as the water underneath the bubble rebounds to take the space left by the bubble, it can jet upward, carrying more organisms into the air. Others have frequently modeled what happens to bioaerosols after they've been emitted, or they've lofted bioaerosols to study the effectiveness of bioaerosol monitoring equipment. But we still don't have a whole lot of information to be able to describe the mechanisms and characteristics that affect whether an aerosol will be formed, or to be able to predict their release. So learning more about this can help us to develop better ways to mitigate bioaerosol release and keep plant workers safe. In the experimental bioaerosol work published at AFIT, we collected aerosols produced in a small, mostly closed reactor with a known quantity of BG colony-forming units, usually about 6 million. Colony counts from the aerosol samples and liquid samples that we collected from the reactor were used to try to calculate a percentage of the reactor population that was captured in order to try to understand how factors contributed to the bioaerosol release. What we found was that with rather modest increases in both mechanical agitation in this top graph and diffuse aeration in the bottom graph, we observed really large increases in bioaerosol capture. In general, we found that bioaerosol capture from mechanical agitation using a stirring bar correlated very well with some of the more physical properties of moving water, like Reynolds number or Froude number. Bioaerosol capture from diffuse aeration correlated better with things pertaining to the air moving through the water column, like the superficial gas velocity or the volume of gas flow per liquid volume. There was no correlation with Reynolds number at all, and there was only some correlation with the calculated Froude number. It was also found through the course of the investigation that the addition of free-floating carrier media in the form of styrofoam beads was able to cut down the amount of capture from the organisms by about 90%, or at least one order of magnitude. So here at EPA, we're continuing this bioaerosol work and expanding on what we know about how bioaerosols are emitted by looking more closely at the composition of the liquid that aerosols are being emitted from to see how the matrix compositions affect the emission and capture of aerosols. Our bench scale system pictured here uses an aeration pump to provide aeration to a relatively closed reactor. And here we have our bioaerosol sampling device connected to a vacuum pump. We began with dechlorinated tap water in our reactor, and then looked at synthetic wastewater, which included those chemicals that are found or that mimic chemicals in wastewater. And then we tested some return activated sludge collected from across the street here at various concentrations with synthetic wastewater. In addition to BG spores, we're also testing these media conditions in bench scale with MS2 and Phe6. So we're bringing the viruses back. The sampling methods for BG also allowed us to monitor the capture of other contamination organisms that we've found, particularly in the return activated sludge, but also sometimes in the synthetic wastewater. These were not identified. We simply counted the number of contamination organisms. So looking at the percent capture of all of these experiments with each organism, regardless of what matrix we're using, shows that BG capture is significantly more, at least more than an order of magnitude higher than either of the other two organisms that we've been monitoring so far. What causes this discrepancy is not quite clear. It may be a property of the spores that favors aerosolization, but it could also be something about the biosampler's construction that favors BG capture over other organisms. That's one of the caveats about bioaerosol monitoring, is that capture efficiency of the equipment can change with the size of the aerosol particles. MS2 viruses are much smaller than the bacteria and are prone to sticking together to form agglomerates. These agglomerates may lead to an underestimation of the percent capture of the reactor. MS2 yields also were not much different from the contamination organisms, even though the starting concentration was supposed to be the same as the BG. So I found that rather interesting. Okay, so let's take that bar graph from the previous slide and split it apart so that we can see what the effect of each matrix type is. And we see some interesting trends emerging in this bar chart. So these matrices are arranged in order from left to right of increasing total suspended solids that we get until we get to a matrix that is composed completely of return activated sludge. For the contamination organisms and BG spores, there is a distinct drop in the percent capture as we get closer to that 100% return activated sludge matrix. This might be expected if the organisms are attaching to the solids, making them too large to be captured by the biosampler. Now, this pattern also made me want to take a closer look at the contamination organisms. In the figure over here to the left, you can see that the number of contamination colonies found in the aerosol decreased radically when the total suspended solids increased, despite the fact that the liquid in the reactor apparently contains more of these organisms. Another detail to look at for BG spore capture is, again, in the bar chart here, just looking at these first two. So we see a drop between the dechlorinated tap water, that's our blue bar, and the synthetic wastewater, our yellow bar. Now, the suspended solids in the yellow bar, in the synthetic wastewater is rather low. So there isn't much for the spores to stick to. As such, it appears that this drop may be a result of the organic chemicals in the synthetic wastewater. And finally, I found it very interesting that despite all of these interesting trends that are showing up for spores and contamination organisms, MS2 seems to be only slightly affected by these changes in liquid matrix. So our working theory so far is that the bioaerosol release decreases as concentration of suspended solids increases. However, we're still conducting experiments to try to fill in the gaps. I'm hoping that more data will help to resolve our uncertainty here, and that we see so far, and let us know if there are other factors at play. This particular figure shows a log percent of capture for individual experiments plotted against their concentration of suspended solids on the x-axis. One more interesting detail that comes from the methods here: these bench scale experiments are run in series of 3 each time I do an experiment. So one reactor containing inoculated liquids is being used for 3 separate experiments. So this would be one group of 3, all done in one day. In the MS2 experiments, especially those containing large numbers of organic substances, the apparent yield in both the liquid and the aerosol samples increased so that each experiment shows a higher yield than the one before it in the same series. These viruses stick together, as I said, but they also tend to get separated by each dose of the aeration, causing the detected aerosol concentrations to increase over time with each new experiment. So our findings so far suggested that suspended solids have an important role to play in bioaerosol release and capture in a biosampler. Organic substances seem to affect MS2 release less drastically than it does BG, but MS2 agglomerates may be more easily separated by the aeration process. This may lead to higher aerosolization yields at a wastewater treatment plant over time for viruses like MS2. For this study, before we can call it finished, we still need to test some more suspended solid concentrations, particularly in that 1,000 to 4,000 milligrams per liter total suspended solid range. We'll also be conducting some pilot scale work in the activated sludge treatment system from the first study to look at the differences between the surrogate's behavior at larger scales. We'll also get to see if MS2 emission is mitigated by free-floating carrier media as well as BG was. We also still have to see what Phe6 does at the bench scale, and I'll be taking a look at an alternate form of suspended solids, which will include potato starch. So with that, thank you so much for your attention and interest. I just want to give a special thanks to the Municipal Sewer District of Greater Cincinnati across the street here for providing all of our sludge and biosolid samples for our tests. I also want to thank the Aptum contractors helping us with this work and my co-authors. With that, here are my references that I've used throughout this talk. And with that, I'm happy to take your questions.
Thank you, Adam. That was a great presentation. And we do have quite a few questions, so we'll go ahead and get started. The first question is, during the activated sludge treatment system and discussion on slide 9, what about using somatic colophages as surrogates of enteric viruses? Hopefully I said that right.
I can certainly see how that might help. There's to some extent, I sometimes wonder how much this is related to a particular type of virus, and how much of it is simply a matter of we need something that we can track easily and is about the same size and has roughly the same composition. But I think one of the main reasons why we selected the surrogates that we did is partly because they are easy to cultivate, they are easy to monitor, they don't typically occur naturally in the synthetic waste or in the sludge. They don't occur naturally in the return activated sludge, so that makes it a little easier for us to find them. And it also they also have a relatively easy method for analysis. So we'd have to look at all of those factors: the method of analysis, how easy they are to track, how good of a surrogate they would make for whatever we're looking for.
Right. Next question is, did you see the activated sludge bench top set up with sludge from a full-scale plant?
I'm sorry, repeat that again.
Yeah, it's a little. Did you see the activated sludge bench top set up with sludge from a full-scale plant?
I'm having a hard time understanding what it's asking for exactly. I mean, when it comes to downscaling something like this, there are a number of challenges that you run into, and I'm not sure if it's as useful to bring it. When I think of bench scale, I think of something that can be set out on the table in front of me. The system that we have is more of a pilot scale, and the problem is when you shrink these systems down so much, it becomes you're taking it further and further away from the full scale. So if you can run it in pilot scale, it's a little bit better. I don't know if that covers the question or not.
Thank you, Adam. We'll see if we get a follow-up.
Yeah.
Next question is, for viruses, are plaque assays or quantitative PCR used for titering?
In this case, it's going to be plaque assays. We're looking for viable plaque forming units.
Thanks. Next question is, what is the lab temperature range for room temperature?
Typically between 20 and 27 degrees Celsius. Probably closer to 23 to 27 degrees Celsius. I'm being somewhat conservative in my estimation there.
Great. Next question is, at which level of log removal do you need for a biosolid water treatment?
Well, obviously more is better. And so let's see, I think if I'm remembering right, it's been a little while since I've looked at the difference between the Class A and Class B biosolid producers. But as I recall, you had to get at least one or two log unit removals for one or two log units of removal for Class B. And Class A is supposed to be considerably better, close to complete removal, if I'm remembering right.
Great. Next question is, regarding conclusion that BA may colonize a water treatment system, BA spores are very stable in environments, but don't readily germinate unless it's introduced into a host, unlike BG, which likely has an environmental life cycle. Do you have evidence that BA germinates in water treatment plant matrices?
That's a really good question, and that would require some more work, I think. So yeah, and that's one of the things that I think has been generally considered to be a criticism of using some of these surrogates, is that sometimes they don't behave the same way as the original organism, and unfortunately, our current setup is not set up to be able to do something that would be more close to what we need. What we would need is something more like a BSL2 setup for the pilot scale, so that we could use something like BA Stern strain, the vaccine strain, for instance. But that is one of the common concerns about using these surrogates. So that I would say that's a valid question, a valid concern, and maybe there would be a way for us to explore that in more of a bench scale, something that could be conducted inside the biosafety cabinet or something like that. So that's a good question.
Thanks, Adam. The next question is, can boiling water at a water treatment plant be feasible towards eliminating pathogens, viruses, etc., from treated water?
The biggest, the biggest issue that holds that back, I think, would probably be just the sheer volume. I have to take a look at the volume of water that's treated at over here at MSD, but it's in the millions of gallons each day. So you're talking, if you have contamination, a contamination event of inflowing water, I'm not sure that boiling one of those large aeration tanks worth of water would be a viable option.
Okay. The next question is one of your hypotheses that the moist conditions make BG more vulnerable to dry heat than dry conditions?
Well, the moisture would cause the. I think there might be a little bit of a typo in there somewhere. The moisture should make it so that the heat is moist heat. At least that's how I understand that. What we originally were thinking was that the heat from like the incinerator, for instance, would drive off that moisture before it would even have a chance to contribute to the inactivation, or we figured that it might even protect the spores, protect it from and create some of these cool spots.
Thanks, Adam. The next question is, what type of method did you use to measure the phages on slide 27?
So let's see here. Slide 27. I'll make sure. Slide 27. That was not a.
It might have been for an earlier one, sorry, but around there.
Oh, I see. I believe that for all of our bacteriophages, it's been a single layer auger method. Let's see, and it uses an EPA method for cultivating bacteriophages. So it's definitely one of the SOPs. For the first study, I'm not sure if it was a single layer or double layer for MS2. I'm not as familiar with Phe6 cultivation, but certainly for the later ones, the bioaerosol work that we've been doing with MS2, that's been a single layer auger method.
Thanks, Adam. The next question is, are there any studies that investigate the fate of pathogens in wastewater collection systems prior to the entering the wastewater treatment plants?
As in where? I'm guessing that pertains to how the pathogens would actually make their way into the treatment plant itself. Not that I've seen. I'd have to go digging through the literature for that one.
Okay. Also, another question from that individual is, based on the findings, could you please comment on if there are any precautions or level of PPE wastewater owners should consider to prevent potential risk of contact with pathogens?
Well, I mean that's kind of going on the edge of suggesting courses of action and getting into policy. And I'm just in the field of research and development, so not necessarily that's not exactly in my wheelhouse to actually prescribe any kind of methods to protect anyone. I mean, if I were working closely with these materials, I would probably just use the same kinds of PPE that have always been used in laboratories: closed-toed shoes, the lab coat, safety glasses, those kinds of things.
Great. The next question is, any thoughts on using pressure to help gain your higher temperatures?
That would be something that would be easier to do at the you're talking about in the composting, I'm assuming, and that would be something that would be done more likely at a larger scale than these small composters. I would want to see what the pressure is for a large scale system, because otherwise you're running the risk of just turning it into a pressure cooker. But even so, most composting setups, you're going to have a place where the material is not under that kind of pressure. So you still have the same kind of problem with getting the target temperature, target pressure, target conditions for inactivating any of these organisms.
Great. In regards to incineration, can you comment on what type of sand is used?
It's an olivine sand, so it's a little bit higher up on Bowen's reaction series, a higher temperature threshold needed to melt it. It does do some interesting things when you incinerate it, though. It has a couple of points in time where the olivine starts to rearrange itself chemically. Once at about, what was it, 600 degrees, I think, and then again at around 800 or 900 degrees. It converts from olivine and ferro and magnesium silicate over to something that's more of like an iron oxide. And at the highest temperature, it actually turns into more of like a magnetite. And that doesn't last very long. That magnetite tends to reactivate or activate back down and oxidize back to something like I believe it's called maghemite. It's like a hematite type of mineral. We're digging into my geology background there.
The next question is, in your compost experiments, did you analyze if the biomass pine dust mixtures met the recommended carbon to nitrogen ratio for aerobic composting?
No, we did not in this case. The biosolids kind of present a little bit of a challenge there.
The next question is, have you ever thought about using a dye-based viability assay versus a plaque assay?
No, can't say that I have. That might be an interesting way to go for that. Certainly might make enumeration a little bit easier.
The next question is, what causes MS2 to aggregate?
Well, basically most anything that's at the nanoscale will tend to do that. When you're dealing with something that's that small, there's any number of different forces that might play a role. You can have hydrogen bonding, you can have van der Waals interactions. They're just when you're dealing with nanoparticles of any sort, aggregation is just going to happen unless you specifically treat it in a certain way. And we didn't treat these in any particular way to make sure that they don't, simply because we're trying to emulate what would actually happen in the wastewater system. An influx of a viral particle is not going to come with some kind of an anti aggregate compound to make sure that it doesn't form some sort of a cluster. So we didn't include it.
Thanks, Adam. The next question is, can you refer to any papers addressing cellular interaction with agitated water molecules? And maybe this person might just need to email you, but if you know anything off the top of your head.
So, I mean, we addressed it a little bit in the bioaerosol review paper that we published in 2011, or I'm sorry, 2021. But yeah, you'd probably need to email me directly to get some information about that, because I can't think of any of that off the top of my head right now.
Thanks. The next question is, are impacts of UV treatment also being looked at in relationship to the survivability of the pathogenic organisms in the research?
Um, not in this particular case. We're just focused on we were just focusing on the things that are normally already found in the in the wastewater treatment plant. We were not dealing with the application of UV. It was just beyond the scope of what we were looking at in this particular case.
Thanks, Adam. We do have a comment here about the wastewater being sampled prior to arrival of the treatment plants. The comment is that there might be some examples in the COVID tracker data because they were trying to trace viral sources back to smaller and smaller sewer sheds.
Yeah,
so
that makes sense.
The next question we have is back to the sand. What happens to the sand after the experiment? How is it disposed of?
Let's see. In this particular case, it was autoclaved, sterilized, and disposed of that way to be thrown away. We didn't really do much with it beyond that.
Thanks. The next question is also about sand. If the same sand is used at large scale for wastewater plants, what would be its expected lifespan?
That's another good question, and I don't really know how long it would last. I know that it is the same material. I don't know if it's the same grain size. Back here at MSD, the lagoon looks kind of red because of all of the olivine sand that's been used up. And that would be probably a question for MSD to ask them how long they keep the sand in there, how long it takes before they switch it out. We only used it for one experiment each time.
Thanks, Adam. And one last question is, what effect does the alpha pinene from the needles have? I said that correctly.
Needles? We weren't using needles.
Then I don't know. Well, that was the last question. Maybe that person can email you as well if they have further questions. And if anybody else has any questions, Adam's email is here up on the screen. Thank you all for joining us. We appreciate it, and we will see you all next month.
Thank you.
Thank you.